try and keep this moving pretty swiftly. My name is Nikesh. I did my PhD here in bioinformatics in Gary's lab. And just like one of these themes in the Nolan lab, you never really leave once you're, once you're done. So since then, I've done a postdoc, and I teach a couple classes in computational immunology and informatics while I'm here. One of the primary things I do now is work on Cytobank, which is a way that we designed to kind of work with all the data that's been generated, especially in the Nolan lab, and more recently with all the mass cytometry data that's out there. I'm going to kind of walk you through what's there, why we did what we did, and kind of show you a couple things. This next couple days will be around to kind of walk you through what's here. This is also a way you'll be able to get all your data to take back with you, as well as a lot of resources that we've built to kind of highlight and really make flow accessible to a variety of people. So just before I start, there are a couple of people from Cytobank here. So Angela, who's in the back, and Stu over there. As we're going to going around, just please feel free to ask us questions as we go through it. Okay? So kind of a question of why are we doing this. So when I came into the lab, I came in with an informatics background. I really hadn't done any flow myself, but then I got here and Gary had me go to the lab right away. And then with Peter and Jonathan, I did a lot of flow. So I did my own collection of data. I did my own analysis. I spent lots of time at the Caliber and the LSR2. And since then, I've had to deal with a lot of the CyTOF data coming out. And one of the things I ran into when I was trying to do this was, I didn't come in to do biology work in terms of wet lab. It was really useful for me to do it. But it was, the problem was that when I started analyzing the data and came up with some new ways of looking at the information or trying to deliver new ways of visualizing the information, there was no easy way for me to do that. What I needed to do was have people send me their FCS files, along with that some information about what those FCS files were, and then maybe they need to tell me how to gate the data, and then maybe I'll take all of that information and return them a slide that shows some kind of a result. And we have this conversation over email of how did you gate the data, what does this mean, why did you do this, et cetera, et cetera. Anyway, that didn't scale. And Peter and Jonathan and I started sitting down and trying to think of, okay, what do we do with this data, and how do we really get it accessible to the computational and statistical communities? And that's kind of how Cytobank started. So I think all of you are here. I mean, I'm really excited with the poll Peter did this morning to see how many of you are doing really high color flow cytometry. As you can tell, we're biased. We think that this is a great tool for single-cell proteomics and has a key impact in what's happening out in the field today. Again, the idea of being able to do the phosphoflow, to me, Matt Hale used this analogy before, was really kind of this idea of bridging biochemistry and immunology, right? In one assay now, you're getting a way to look at cell type, cell signal, cell cycle, and all these features measured at the single-cell level. So how do you take advantage of that, and what do you do? And then, as we know, we're doing a lot of work with stem cells and regenerative medicine. So what do you need when you start doing a lot of high-dimensional data and start thinking about this information in a systematic way, right? Like we're going beyond the 2, 4, 6 color assays everyone's running. Very quickly, you realize you need to kind of have a place where your FCS files are backed up and managed, right? They can't be sitting around in your desktop anymore, just in whoever's folder happens to be the computer for. The other thing, again, this has happened to us so many times, right? What the heck are those FCS files? What did people measure? How did that happen? What kind of details are there? So can we get that information along with the plots that someone's doing to be able to show the results? And then a variety of ways kind of looking at that information. The other thing, it's happening a lot in the Nolan lab, and I'm sure as you guys are doing more of this high-dimensional cytometry, you're seeing this, is that it's rare now that you're analyzing the data by yourself. Usually it's with 1 or 2 people, maybe a team of people, and these people tend to be not just folks from the lab next to you or a collaborator, at another lab in an institution, but also across disciplines, right? So you're talking to folks in statistics, computational biology, informatics. How do you get them to data the way they want it at the level they need for the information? And then finally, the next one is this platform, right? So again, how do you become yourself not be a bottleneck for trying to do this? So, large computations and visualization, some of the things you guys are going to see over this next couple of days, you'll see kind of the need that's arising for a lot of large computation information or a way to deliver visualizations. So how do you do that without really worrying about where someone's coming from, right? Whether it be their iPad, their desktop, et cetera. And then finally, this one kind of comes up over and over again, is that if you're a PI or a manager and you're working with your grad students, you guys are doing some great analysis, you kind of come up with a paper, you kind of get it out there, and then your grad student leaves because they got the next faculty position, et cetera. And then a few months later, the paper reviews come back and you need to follow up. What do you do, right? Because the grad student's taken their laptop with them, all the analysis has gone along, and it's hard to kind of follow up. And so how do you kind of make sure that your data is there in a way that you can follow up and transition as needed, especially in an academic or in an industrial environment where there's a lot of transition with this information? So kind of a quick snapshot of Cytobank. Again, we'll get into more of these. We're here to answer questions. One of the things I really like about this class, and I really encourage you guys to take advantage of, is that it's very rare to get access to the folks here in the way you guys are going to in the next 2 days. And the lectures are nice, but it's the conversations you're going to have in the afternoon today and tomorrow as you talk to these people that are really going to kind of drive home some of these points. Take advantage of that and ask lots of questions. But anyway, digression. Basically what happens is you have a collection of FCS files. We call it an experiment. You upload them to the servers, and essentially all you do is everything through a web browser with a connection to the internet. That's all you need. You can manage your data using something called the experiment inbox. And I lost my, okay, there we are. And through the inbox, again, this is very similar to what you would look at with your email inbox. Each row is an experiment that you've either uploaded or someone has given you access to. And within there, you can see a number of FCS files, illustrations, who the primary researcher was, things like that. And then there's a way to manage and organize all of this, either using something called labels, very similar to folders in your email system, or the annotations you've made in the experiment. And then there's an entire system of collaboration and security built in, so projects, et cetera. And then if you click on a particular experiment, you get to the details page. Within the details page, you can see information about the FCS settings, the machine stuff, all of that. You can share with someone very quickly, so long as they have an account on the Cytobank. And you can download or clone the experiment as needed for a variety of different analysis. And then when you want to do analysis, you would click into this illustrations tab, and that takes you to an entire couple sets of web pages here that let you analyze your data. So through this interface, you get to annotate the information, you get to gate all your data, you get to generate heat maps, contour plots, histograms, all of those types of things. You can also do advanced things like SPADE. And the nice thing about all of that is everything's linked back to the underlying data. So as you mouse over a heat map, you can see what the underlying plots look like. As you do a SPADE analysis, you can see what that information looks like. You can see what the 2D dot plots look like. And again, as everything's linked back, when you do share with someone, they can just see the result you have, but they also have a way of seeing how you got to the data that you got to. Okay? Any questions on that? Yeah.
So does this require that your FCS files get uploaded to the
Yes.
server?
Mm-hmm.
Because sometimes these files are pretty big.
Yeah. Yes. Correct. So it requires that the FCS files get uploaded. But once the files are uploaded, all the analysis is happening on the server. So all you're getting back is web pages. So the initial time when you're uploading your data, you may need a fast connection. So we've seen anywhere from hundreds of megs to gigs of experiments being uploaded. But then again, we're not sending anything back unless you specifically download the data. So that's usually kind of the way this works. Very similar to like once the data is there, kind of what you do with Google and Facebook. You're not worrying about how the compute's happening. You're just getting these web pages that are looking at them. Yes?
How does it deal with data when, say, Diva compensates one way, you do manual compensation in Flojo? How does it deal with that?
So if you've compensated in Diva, so the question is how does it deal with compensation in terms of the different ways that are there? So when you upload the data from Diva, for example, and you've compensated in Diva, that compensation matrix is actually part of your FCS file. And so by default in Cytobank, you can say, I just want to use the compensation matrix that's in the FCS file and continue doing analysis. If you want to, you can recompensate the data. So there's a built-in compensation and ways to do auto-compensation in Cytobank. Or you can say, I've done my compensation in Flojo, I want to import the matrix in, and then you can adjust it. You can also just take the file compensation matrix, adjust it. Say, I want to adjust the file compensation matrix that's there, and then apply it. Okay. Any other questions? Nope. Okay. So just very quickly, how do you get to Cytobank? So we have a free version at cytobank.org. I think many of you registered there today. and that gives you kind of a lot of the base functionality that you're seeing. We have a version for the mass cytometry community at the DVS cytobank that gives you actually access to some premium modules like SPADE, and there's a free 30-day trial, so you can just sign up and start using it if you ever wanted to do kind of SPADE out of the box. And then we have enterprise versions that we get to put out for a lot of the companies or large labs or facilities that want to kind of have their own cytobanks for a variety of reasons. I think as kind of mentioned, and you'll hear a little bit on the talks, a little bit from Sean and some of the talks tomorrow, we kind of by default became the platform to deal with all this data. And this happened with just the fact that all this information was being generated here. And it really underscored kind of our approach and the things we were trying to do. It's because the landscape changes as soon as you start generating 30 or 40 parameter data in terms of how you deal with it and what you're thinking about, right? You don't want to look at the two-dimensional plots all the time. So, but in addition to CyTOF data, we deal with a lot of flow data in general. So what excites us a lot is if you look up, there's a URL down here you can go look at, even in the past couple of years, we've seen over 60 or 70 publications that have come out where people have used us in one way or another and across a variety of range of applications. So, so it's really exciting to actually see that. So how did we get started? What happened? And the reason I like to tell this story, because one thing I think that flow is, again, it's very powerful, but it's sitting about 10 years behind in terms of computational approaches if you think about microarrays and next-gen sequencing. And really kind of the approach we've taken here is to kind of accelerate that. So can we get to a point where we can start seeing flow being applied in these types of areas, or really kind of the questions we want to be able to ask? I think the context you get with flow is much more powerful. So the story actually starts out very simply. So this is a data set from Jonathan Irish. It's published in Cell many eons ago now. And what he had done was he had looked at, this is caliber data. So again, we're not even dealing with 30 parameter yet. He had looked at 6 parameter data. He was looking at 30 patients across 6 conditions. He looked at about 6 different phosphoproteins. And all of a sudden, he had 540 files he needed to condense down into one figure for his Cell paper. How do you do that? And what do you kind of see? And this paper is actually one of these landmark papers that was the first ones to actually show that signaling is clinically relevant, especially when you take a look at leukemias. And this is a very interesting and a really powerful approach to be able to bring that information in. And so what he did was he actually borrowed from the microarray community. He said, okay, I can make a heat map. I can summarize the median values. That kind of puts across my, my summary of the fact that I can see diseased versus non. But as we all know, that there's a lot of underlying information that was being hidden. But on top of that, when I came in, the point was, okay, how do we actually do this so that we can generate a heat map and be able to mouse over and see what that underlying data looks like? Because we didn't want to lose that information, right? Because it was very powerful. And it was kind of like a be careful what you wish for type thing, because all of a sudden we realized, okay, heat map making is not hard. Mousing over is not hard. But in order to actually do everything else, I needed to know what the FCS files were. I needed to know how someone gated. I needed to know what was measured. All of that information then kind of had to start coming in a way. And then, again, if we have all of this, we want to be able to go back and forth so we can kind of zoom in and focus in what's happening whenever we wanted it. And so that kind of took us down this road of trying to build cytobank and getting all of this information in one place, et cetera, and has led us down to here. So you can blame Jonathan if you guys know him for what's happening here. And again, why was this a hard thing? So if you look at how flow assays work right now, right? So you have a sample, you run a flow assay, you go through a flow cytometer, you get some information in your FCS files, and then you'll go through and you'll do your gating somewhere. Maybe you'll do some advanced analysis in another place, and then finally you'll make a figure, right? And as you start doing this, you realize all the information that you need to do this collaboratively or to actually do this in a scalable way gets set up in a variety of different places, right? So some information will be in your lab notebook, some information will be in FCS files, you'll have your gating information in one place, your statistical information in another, and then finally you'll make a PowerPoint or a PDF, right? And then as soon as you start thinking about having to go back, or your PI asks you about how did you do X, Y, Z, it becomes to be very annoying, right? And this is what is happening to us. Gary was asking us all these questions. It was getting really annoying to answer, so we started building this stuff, right? And again, this kind of then underscores what happens, right? So all of a sudden, this is one FCS file. You start looking at the data in one FCS file. You're going to get a look at 2 to the 32 plots or whatever definition that is. And the other thing that's happening, right? It's a team of people that are analyzing the data. And on top of that, it's not always just about design and bench work. That is still very important, but you're going to realize very soon, as soon as you start doing more of these experiments, that you're going to be spending a lot of time in analysis. So having a way to do that in a standardized way that is accessible to a variety of people becomes really important. And this essentially says the same thing. And so what we started to think about, and again, this is turned out to be much harder than we thought. We're still working on a lot of stuff, but really the idea is that we want to be able to go down this path, right? So have figures linked to experiment details. So depending on whoever you are and whatever you were doing, you had a way to focus, right? So if I'm talking to my PI, or a clinician, and they're really interested in those results, they can focus over here. But as soon as I'm talking to my statistical collaborators who want to kind of see how I did something or get to the raw data, there's a way for them to kind of be able to do that. So they can go back and forth to see what's happening. And again, sorry, there was an extra slide in there. And in order to do that, we had to kind of take a step back and see, okay, what's happening in flow cytometry and where things are going. And as you can see, there are two things happening, right? So people are increasing the number of parameters. I think a lot of you, at least not a lot, but a fair amount of you have cytophs now. And the other thing that you want to do is you want to start running a lot of samples, right? Like we see a lot of people who want to be over here. They want to run lots of samples on the cytoph with the plates. But I'd say still, even now, a lot of typical flow sits over here, especially in the clinic. And if you look at that information, right, if you're running a few parameters and some samples, there's a lot of really good software out there, right? Because your big problem there is gating. You kind of want to have this way of identifying your cells, and a lot of that's right now done manually. As soon as you start thinking about this area, though, gating's still important, but it's not the only thing, right? You got to be able to think about how to organize this data, collaborate on it, build new platforms. Make sure the annotations are there. So the other thing I always tell people, I really don't care where you analyze, so long as you annotate the data, it becomes really, really useful. So kind of just think of this this approach of, there's kind of this famous child psychology experiment where you kind of get these kids in a room and you say, I'll give you 1 cookie in 5 minutes or 2 after 10 minutes. So to me, like, it's worth waiting those extra 5 minutes for the 2 cookies, and that's really what annotation is. Like taking the time to annotate your data really makes it useful down the road. And so and then this happened to me too. So my work was published in Cancer Cell many eons ago now. It was a diagnostic for juvenile leukemia, and this is what I was doing as I was trying to do this work. I was having to go through multiple applications, having multiple people touch the data, and I realized very quickly, especially with Peter and Jonathan as we were working on this, that this doesn't scale. And then really, again, that's where the idea of Cytobank came from. Okay, so I think you guys agree with me. Cytometry is growing more powerful. Parallel experiments are getting bigger. So we kind of made a decision to put something together to simplify the data sharing and analysis. And again, we're not going to be able to do all the analysis that everyone wants to be able to do, but we want to make sure that people can get to their data in an accessible, annotated manner that's going to make it easy for someone else to be able to do the analysis. And I kind of went through this rationalization of why we built Cytobank and all of this, but really, honestly, it was to kind of make sure Gary could give his next talk, because when he's your boss, you gotta do what he does. So, what does Cytobank actually look like? We went through this. Let me see if I can actually switch over. Let me make sure things are working. Okay, great. So let me I'm going to adjust resolution really quickly. That's good. Okay. So this is what Cytobank looks like when you kind of come in. You'll see here I have experiments that have been shared with me, or people have, I've uploaded or people have shared with me. Just to kind of give you an idea, it's kind of funny, I haven't run an actual experiment, like besides some basic stuff, in a while, but somehow I have about 1,400 experiments in my in my inbox. So it kind of tells you again the idea, right? So there are different ways of being able to get to your data. There's ways to manage all that information through your projects. There's ways to kind of filter down to experiments that you're interested in. And again, this is the idea of annotating your data and being able to bring all of that in, and then being able to kind of organize your data using a variety of labels or folders in Outlook. Okay. When you're ready to analyze the data, again, you're going to click on an experiment that you've uploaded and be able to bring that in. And then you'll be able to do something like look at the details, make some illustrations if you want, be able to kind of come down, click on information like this, see what the machine settings look like. So this becomes very useful if you're working in a core and you're trying to help someone troubleshoot their experiments. You can kind of see what all of that looks like. And then there's places to attach things. So if you want anything that's a PDF, Excel sheet, PowerPoint, whatever else you want to attach with that experiment, there's a place for you to put that. And then there's a variety of ways to do analysis and illustrations and stuff. One other thing that we've added is something called cloning. So let's say I share an experiment with you. You don't want to mess with it. You kind of want to do your own analysis or follow along. What you can actually do is you can clone your experiment. So if I clone this experiment, it's basically simulating me downloading the data and re-uploading the data so that I have my own essentially clean experiment as you go through. So when you upload your data, this is what it'll look like. You'll see something like this where you'll see all your experiment files in one place, your compensation controls in another place, and then down here you'll see essentially plots coming up as we go through this. And then when you click on View Experiment Summary, again, you're going to see data like this where I'll see experiment details about who uploaded it, what was happened, any attachments I have, any protocols. And then if I want to, I can share with someone. So if I wanted to share this with Angela, for example, I would type her name. I hit enter, and now she's getting an email about that I just shared this experiment with her. So by default, your experiments are private, so you're not sharing with anyone unless you want to. But then there are different levels of sharing. So the level I just did is full access, so that gives her full access. I kind of trust her to do all the things. But you can also do things through projects where you can say, I only want someone to just see my results. I want them to be able to work with my figures but not touch my gates, or I want to give them full access. So those are all the levels that are there. Any questions? Yeah.
Can you give an example for a simpler, say a tan color experiment that's been run, in terms of the amount of time that's dedicated towards that data analysis? I understand that's where you're trying to crunch the time or compress the time. Can you give an example of how it's improved percentage? 50% less time, 25% less time. Can you quantify that?
Sure. So the question is, how has the time improved for data analysis for smaller experiments? So the problem with data analysis for smaller experiments isn't the actual, I'd say, analysis part, like gating, let's say. The problem is standardizing and getting all the information together. So let's say I'm running plates of 4 to 6 color experiments, right, which I think we see a lot of people doing now. Along with those plates, you get 96 files per experiment. So let's say I run two plates. I'm going to get 192 of these files, right? Along with those files, you need to have kind of the information of not just how you gated the data, but, was it a titration? What compounds did you look at? What else did you measure? All of that information kind of needs to come in. And then you may want to be able to then say very quickly, give me a broad view of the data. So can I see a heat map of my plate really quickly, and as mouse over see what the underlying data looks like? So all of that kind of comes in built in over here, where there's a place for you to systematically add this information in, and then be able to share it with people. If you've actually annotated at the cytometer, this part becomes really easy. If you haven't, then there's a way for you to do all of that through the interface. So you're not going to save much time if you're running 3 files on a 4-parameter experiment, let's say, but you're going to save a lot of time if you start doing 20, 30 files in a 4 or 5-parameter experiment. But the big thing, even if it's a 3-file experiment, is that if you have it in this place in a cataloged manner and you needed to go back to it 4 months from now, there's a way for you to do that and be able to see what's happened. Does that answer your question? A little bit. Okay. So this experiment I'm just going to walk you through right now is, you'll see here, it's actually a 5-color experiment, or a 4-color experiment, CD3, CD4, CD8, and ERK. So you'll get a sense of how quickly or how not quickly this gets analyzed. Does that help? Okay. Any other questions before I continue? Nope. Okay. So this information sitting here, again, what I'll go ahead and do is I'll click on my illustration. And I'm skipping over a lot of stuff around management and organization. We can come talk to me a little bit about that later on. The key thing we try and do that again is different from other analysis programs is we try and ask you for a little bit of information about your flow files ahead of time. And if you can give us that information, then the process of analysis becomes easier because you use that information to organize your plots and be able to kind of drill into the information that you're interested in. And so in this case, the experiment I just uploaded is a pretty simple 4-parameter experiment: CD3, CD4, CD8, and a phosphol. And I'm looking at PBMC across a variety of different conditions. So very similar to that experiment you guys are going to do this afternoon in the lab. So when I uploaded the data, the first thing I have to do is kind of look at this area called figure dimensions. And there are two things in the figure dimensions you have to always tell us. One is, what did you measure in each channel? And then two, you have to gate your information. So if you have to do any gating, then you do it through something we call populations. And then depending on the experiment you might have conditions, doses, time points. All of those are available to you. We'll be using conditions today. And then if you decide you really don't want to annotate or that really isn't very great, there's a dimension called FCS files that just gives you a list of all the FCS files. And then if you ran this as a plate, your plate column, row, and plate dimensions will all get annotated. So there's a lot of stuff that's automatic, and then things built on top of that. So the first thing someone has to do when they come in here is they would come into the channels. They would click on this button called Setup. And then down here, you would see a couple things. So you would see my files. Again, I have two different sets of files. One file I ran CD3, CD4, CD8, and ERC. And another file I ran CD3, CD4, CD8, and STAT3. And so again, automatically, based on what I'd annotated as a cytometer, the files were grouped. So this is one of those where I would love for people not to have to come in here, but people don't always do this. And so this is an opportunity. For example, if this was ERC and I wanted to call it LXS 647 as opposed to 647, I could do that. All the data that you upload, we do not touch, so you can always get back to the data that you originally uploaded, and then everything else is done as a layer on top of the information that's over here. If, for example, that you had done this and maybe this is a caliber and something got messed up and you needed to move between panels, you can move between panels, and you can also create your new panels as you needed. For most people, they're going to come in here, they're going to verify that everything looks good, and then they're going to click Return to Illustration. The next thing you may do is you may click on Populations and you'll click Gate. And then when you click Gate, you go to an interface like this. So the two requirements for being able to use Cytobank, because you need a web browser, a connection to the internet, and you need the ability to run Java applets. And so the two places we use Java applets is for uploading your data and also for gating. So those are the kind of the two things that are going on. Yeah, it should be fine. I tested this morning. So, okay, I'll switch over to another one. My computer is going crazy. Okay. Let me just switch over to another one. Sorry, I just actually ran this right now and I did something to my computer. Oh, there we go. Okay. So, yeah. Okay. This has been okay. All right. So while that is coming up, I think I should be able to do the rest of them. I'll get to show you gating in a second. Let me just go ahead and finish up some of the other stuff that's happening. Why did I do that? Okay. So let me just finish up and then I'll come back to that. So once you've done your gating, the other information that you need to be able to put in is things like different stimulations you might have. In this case, again, I did this experiment and I looked at unstimulated PMA, IL-2, IL-4, and IL-6. And again, what we're going to do is we're going to go ahead and look at the information that is in your FCS file name or in the sample names that you're looking at. And then based on that, you can essentially enter in a variety of different conditions or codes, and we'll match based on that information. So in this case, I just went ahead and I typed in unstim, and it went through and matched based on file name, sample name, etc. If I didn't do this, I could, or if it didn't match correctly, I have a way of changing this information as I go through and assign it in different ways. So similar thing for doses, individual sample types, and so on. And let me just go ahead and reload this to see if it will come up. It should be okay. Oh, there we go. Okay. So, and then essentially how I do gating, again, is there's this interface like this. I can look at my data as forward versus side scatter, or I may change it to side scatter versus CD3. I can go through and look at different versions, different files as I go through here. And then what I'm just going to do again is go ahead, say forward versus side scatter. Maybe I'll draw kind of an intact cells gate. And then within that intact cells gate, maybe I'll zoom in and then go ahead and draw essentially CD3 versus side scatter, let's say. And then within CD3, I'll go ahead and draw CD4 versus CD8, let's say. The gates should be probably a little cleaner, but I'm just going to go through and use this as a way to demonstrate what's happening. So I'm basically going through gating my data. You'll see a few things. For example, I'm using the compensation matrix that was in the file. If I wanted to, I could have used a compensation matrix that I had generated before. The populations I've made are sitting over here as ungated intact cells, CD3 and CD4 positive populations. The other thing you'll notice that right now I'm looking at my data CD4 versus CD8 zoomed into my CD3 positive population. So if I wanted to kind of see what this data looked like a one level up, so kind of like a backgating type thing, I can just switch the active population to be intact cells, and you can see what that information looks like just as you go along. In terms of plots, I can go ahead and change it to be a contour plot, density dot plots, et cetera. Those are all, again, standard flow cytometry plots that you're looking at, as well as being able to look at my populations. So one key thing about populations, population is a combination of gates. So that's another key difference that we have in Cytobank. So my CD8 population that's sitting over here is my intact cells gate, my CD3 positive gate, and my CD8 positive gate. So that's kind of if you guys used CellQuest before, that should be a little bit familiar. But again, this idea, and this becomes really useful as you start doing barcoding, because barcoding you might have 3 gates and you combine them in a variety of different ways as you go through it. Gates are specific to the compensation you're looking at, the transformations you've done, and the axes you're looking at. So if you're ever wondering what a gate looks like, you can click on it and it'll just go through and show you exactly how you drew that gate. Also, gates are global by definition. So initially, I can apply that. When I draw a gate, it's applied globally to all my files. But if I need to change it for a particular sample, for example, I can tailor it. So I'll still call it CD3 positive, but it'll look different for one gate versus one file versus another. So that's kind of this idea of global versus tailoring. And then if I'm interested in seeing how this gate looks like in all my samples, I can click check gate, and then down here you can see how that gate applies to all the files that I have in my experiment.
Now I'll ask you.
Sure.
Obviously, I can see it's 100 times better than Flojo, but the problem with Flojo is you're in the middle of analysis and you just have not saved and it crashes.
Right.
And you use all your analysis and you have to start all over again.
Yes.
So how stable Cytobank is?
Got it.
Do you have to save as you go along, or is saved?
That's a good question. So the question is, how do you deal with the fact that your browser may crash or something's happening, and so what happens in Cytobank? So a couple things happen. All the data is on the server. So anytime I'm hitting refresh or upload, all that data is actually getting saved. So the exact last step you did is where we are. So I'll show you that in a second when I'm in illustrations. But that's the nice thing is like I can stop doing what I'm doing, go away, open another browser on another computer, log in, and I'll be exactly where I left off. The one place that isn't exactly true right now is in the gating interface. So I'm going through doing my gating. You'll see there's a button, save and return, whereas everywhere else in illustrations, there's just a return. So when you're here, once you've drawn your gates, you hit save and return, and then you're going to be back into the illustration view. The other thing that you'll notice is there's this area called import gates. And again, this is true for all the other ones, import conditions, doses, time points. So the idea with something like this is that you can, let's say you do an experiment, you've gone through the gating, you've saved it, now you do another experiment that's very similar. You may just turn out your compounds or something. You can come in here, import gates from that experiment, and then start with that to change things as you go along. So as you start doing larger studies or kind of have your template experiment, that becomes very useful in terms of having one thing that everyone's starting from and then copying, see what's going on. Again, I'm not going to go into compensation right now, but there's a way to do auto comp. There's a way to kind of bring in the compensations. All of that is there. And we have whoa, what did I just do? Okay. We have a pretty good support website and an active blog that kind of goes through all this information, including, you'll see over here, there's like YouTube tutorials and things like that. We also have a chat room you can come into and bother folks like Angela. Okay. Any other questions on this end? Yeah.
Can you open multiple gates of the same sample in the same window? Like sometimes to clean up the population, we always backgate. Like
Right. Yeah. So so the question is, can you open multiple populations in multiple gates? You can't do that quite yet. That's coming probably in a month or so. I'll show you an example of where we're doing that with Spade in a second and how that translates as we go forward. Yeah.
Can you do some, I know you can do some pre-analysis and gating before you actually import the file here. So can you save that? Like, can you save a Flojo file with some pre-gating and then import it to do the downstream analysis here?
Sure. So the question is, can you import in analysis from Flojo and then continue here? Right now, you cannot do that because the gating standards are not the same between what Flojo talks and what we talk and what other analysis programs talk. There is a gating standard coming out that everyone's pushing to, and we're actively exporting to that gating standard. So the hope is that we'll be able to do that later on. The process, the workflow right now is you may do some pre-analysis in Flojo, export out your FCS files, and then upload those FCS files and continue doing stuff over here as needed.
So is that standard something that would be in, say, standard flow acquisition software, like Diva, with those gates as you're gating on the fly, be saved, but could then be brought into Sediment?
So the question is about the gating standards and whether those would be standard gate descriptions for like Diva, for example. And yes, so the idea is that this is a standard by ISAC. It's called gating ML. And you should be able to, the idea is that people can export to that standard and then other people can import from that standard because then there's a common way to talk about gates. Getting BD to implement that in Diva is another whole story that we can talk about some other time. But again, that's the idea. So kind of let me get through this pretty quickly now. So how does this work? I just spent a lot of time doing a few things in Cytobank, but hopefully this part of it doesn't take you too long, or as you get familiar with it, this part goes pretty quickly. Because the power, again, is not in gating your populations, it's really in trying to understand what's happening in your experiment and where things are. So that, again, is where these figure dimensions come into play. So what I can do now that I have this information in here, so you can see things like conditions, populations, channels, is I can drag and drop these things around, these dimensions around, and I can choose a variety of things that I'd be interested in looking at. So for example, in the channels case, I'm interested in the two phosphos I measured. So I measured phosphostat 3, and I measured phospho-ERK. Right? And so you can see over here I have phosphostat 3 and phospho-ERK. And then maybe I'll say, I'm not really interested in my ungated population. I'm interested in my CD4 and my CD8 cells. And every time I click update over here, something's happening down here. So you're going to see right now this tube layout of my files. Right? And they're organized in a particular way. So you're seeing, again, a column for every condition I'm looking at, a row for every phospho I'm interested in, and then I'm looking at a table for CD4 and CD8. And the way that's being defined is using these dimensions right here. So you can see here columns, rows, and populations. And so if I want to change things around, I can just move these around, and what I'm actually doing is pivoting my figure. Right? So every time I do that down here, I'm rearranging my figure so that I can see what that looks like. So now again, I made a column for every phosphol I'm interested in and a row for every condition I'm looking at. And you can see what that looks like over here. And if I actually click on illustration, I see plots as of the stuff that I'm looking at. And the way I define my plots is on the left-hand side here, you see plot control. So I can see plot type, density dot. So let's say I want to make them contours. And then you'll see stuff like, what do you want to see on your y-axis? So I'll say y-axis is forward scatter. On my x-axis, I say, well, use the values I chose in my channels dimension. And then things like show gates, show plot statistics, those are all here. And then every time I'm clicking on this green button, click here to update, it's actually updating the server, and that's where I'm saving my last state. And then when I look down here, you can see things like forward scatter versus ERC, forward scatter versus stat3, unstim, PMA, IL2, etc. Now, this is a very simple experiment, right? I have 6 conditions. I looked at 2 phosphos. I have maybe 3 populations. And what you're now doing is you're really just kind of focusing in on whatever you want to look at, whether it be CD4 cells, CD8 cells, what signaling you want to be interested in. So you can imagine kind of just extrapolate. You have a CyTOF experiment. You have 30 channels. You have 10 populations, 12 conditions, 4 individuals. You can kind of see how this could be very useful in just kind of filtering down to what you're really interested in, right? And you can also have built-in things. Like if I want to make an overlay here, I would just say plot type, histogram overlay, click to update, and then down here, you're going to see essentially that information overlay. And I can see very quickly a few things. PMA is hitting ERC, and IL-6 hitting stat3, and then also I have some kind of a population in my CD8 cells that I probably need to go and gate a little bit better. Does that make sense? Any questions? Then I can go ahead and save all this information as needed. There are a variety of ways of exporting the data. So you can export all the events, you can export the gated data, you can export the statistics. There's ways to do all of that. If I say, okay, this is very interesting, I can save it as histograms or whatever I want to call it. I hit save, and now I've saved all of those settings and how I got to that information sitting over here again as an illustration. And again, one of the things I'll point out is if I click on print view, let's say I share this with my PI or my collaborator, and they come in and they're like, well, what did you do? So very quickly they can see, okay, I measured ERC and stat3. I looked at the following conditions. I calculated a statistic in a particular way. I looked at my CD4 cells, my CD8 cells, and then down here I can see exactly how I gated this data. And yes?
I can't see the scale. I mean, obviously I know it's like 10 to 1, 10 to 2.
Yeah.
Because some papers may not accept, they want to see it, so how can you get that?
So the question is the scaling, and sorry, I was trying to get through some stuff. So how does the scaling work on the gate? So you know, right now it's trying to do a scaling for all the median values that you're looking at. And what you would do actually is, if you wanted to, I could click on it, hit OK, and then down here, I have something called plot statistics, where I'm setting the scale. So right now I'm looking at the median value of the scale. So maybe I want to do a full change, let's say. And I'd say my control is my first row, which is the unstimulated. And then I can actually set my scale either manually using user-defined scales or globally. And then when I hit update, it's going to go ahead and change my scales again. So you can see how that information is. So all of this is configurable within the illustration. There's a whole section to do plot statistics to be able to do all of that. Does that answer your question?
No, I meant the scale under the x-axis on the histogram.
Oh, sorry.
Sorry.
Yeah, so that's all over here. So there's
That's
plots.
something you can
Yep.
put in the table, but on the actual figure.
Yeah, exactly. So there's a way to do all of that through the style and scale display as we go through it. So, you know, this is something now where I've actually shared this with Angela, and she can actually sit in the back now and actually draw more illustrations and analysis with me, and I can kind of continue working on things as needed. So it kind of gives you an idea of what's there and how this works and what's available. Okay. So I'll kind of, I was going to show one more thing actually. So how does this become useful? Do I have it open? Nope. Okay. So tomorrow I'll show you how we do this with Spade now and how that kind of comes into play as we go through all of this information. I'm just realizing the timing. I don't want to keep you guys too much. So again, what we went through was a way for you to manage all the data that you had, a way for you to look at the details and put in anything else that you wanted to be able to look at, and then a way to do illustration, where again, if you've done your analysis here, it goes all the way back and is linked. And again, this becomes very important as you start thinking about this new era or new way of doing flow cytometry analysis that's coming up, that's going to require you to have this data in a bit more systematic way. So again, this is just some screenshots showing you how to get the data up. This is again the idea of the figure dimensions, where you tell us a little bit about your experiment. The idea is not to inundate you with telling us every single detail, but give you enough information, give you enough ways to put that information in so you can use it to organize your own analysis. But at the same time, what you're doing is you're giving us, from an informatics and statistics standpoint, the exact information we're going to ask you if we want to do anything advanced with this data. Right? And I didn't show you heat maps, so as you mouse over heat maps, you can see what the underlying data looks like. And then there's a variety of different ways to share this data. So again, everything's private until you're ready to share with someone, and then there's a variety of ways to control that type of access. And so again, kind of just summarizing, and this you can do pretty much, using cytobank.org, manage, share, and backup your FCS files. You share analysis and experiments. Kind of this fear about losing your data, it's kind of in one place. You can do things like remote analysis. So we do this all the time. People, come and ask us questions about their experiments and what's going on, and we'll work with them. We'll say, share your experiment with us. We'll look at it, and we'll work with it, and we'll find out they're sitting in South America, for example. And then the last part is, again, this idea of annotation and capturing this information while you're creating your figure. So you're not worrying about doing that 4, 5, 6 months ago from now. And this happened to me a lot when I started, because I'm not the most organized person. And I definitely had those days when I looked at these FCS files and I was like, what did I do with this? How did I work with that? All of that stuff. And kind of just a little bit of where we're going with Cytobank now is really kind of the idea of a platform for flow analysis, or really for single cell analysis. And the idea again here is to make it so that the data can be accessible in whatever formats and ways people need to be able to do their next questions, right? So if you think about all the systems level questions people want to be able to ask, they want to be able to say, take my flow data and my single cell data, get it to me, and then show me what that looks like with microarrays or Luminex or something like that. How do you actually get this information in a way that people can actually do those types of analysis or ask those types of questions? And what we're essentially doing is building on top of what we already have a way for people to be able to get to this data. And again, this isn't easy. Building a platform is hard. But that's really kind of what we're committed to really, again, get to those next level types of analysis, the next types of things this lab is doing and other people want to be able to do. And so along those lines, we see things like putting in hooks so you can integrate with a core facility. So at the Stanford core facility, for example, there's a button that says send data to Cytobank. And so someone doesn't have to upload their FCS files. They can click on a button, and then we'll take care of the transfer after they log in. Things like doing translational assays and drug screening. So if you're going to start doing things like IC50 analysis with flow data, it can be really powerful. But you want to have captured not just the IC50 plot, but how you got to that population. So as you see an IC50 plot, you mouse over, you can see what the underlying data looks like. Plate analysis, having a way to annotate your data using plates. This, again, we'll talk more about tomorrow, but the idea of how do you deliver something like this to someone, right? So this is a 20-dimensional view of the data. So this is 20-parameter CyTOF data. This is the paper from Sean, I think. And you've gone through, done an analysis, right? You've run SPADE, or you've kind of come up with a way, and you want to be able to use that information to annotate and find your cells of interest. Now, I could come to you and say, install these 5 applications on your computer and go through them to do X, Y, Z. Or I can say, come to a web browser, click on a couple buttons, hit go, and when it's done, we'll send you an email where you can log in and see something like this. And so, SPADE for us is really kind of that first implementation of something like this. What we want to, again, enable is other types of algorithms running with the same infrastructure. So from your standpoint, you're not worried about any of that stuff. You're kind of focusing on the science, focusing on what's happening, and being able to interact and collaborate with the information as needed. Yes?
So if you have information, say you're looking at different patients and you have the same panel that you're applying, and then I want to know what the percent CD38 positive, CD8 cells is in these people versus those, can I export the percentage that I get from the gating in an Excel file?
Yeah, so the question is if...
numbers,
right.
to a graph, if I want to graph them differently, because sometimes with
Yep.
patients you
So the question is, can I get to statistics that I'm generating with flow data that I want to export and be able to use in other programs on a variety of different ways? And the answer is yes. So again, I didn't go and show you that, but you can get to any of the stats you've generated, export it out in a variety of different formats, or you can, which will enable you to do the next plot you want to do, whether in Excel or elsewhere. But also you can then maybe say, you might share with someone and that person might say, I'm not interested in just the statistics, I actually want to see the gate or the data. You can get to all of that information as well in one place. The other thing we do is whenever you export stuff, what we do is we actually attach it to the experiment so that like a month from now you can come back and you can see exactly the data you exported and gave to someone so they can see what's going on. So the current workflow that happens a lot in the lab is people run their experiments, they will upload to Cytobank, do the preliminary analysis and annotation, and then they'll share with other folks in the lab who are kind of going to think about the next analysis. And we may not have the next analysis built in, or they want to do something in R or something like that. They can get to the data however they want to work with it. Yes?
Are there specific clinical data management systems that this is compatible with so you can easily integrate with the lab clinical data?
So the question is, are there specific clinical data management systems that this is specific with? So this version that we're showing you right now is for research purposes only. We are working on a clinical version we'll be releasing later. But the way you can use this with clinical data, and again, you shouldn't be putting in any patient identifiers in your flow data, bad if you're doing that, is you can have a link to your clinical information. So what we do, so for example, the Levy lab downstairs, they have their own follicular lymphoma patient database that the information's in. In Cytobank, they'll have a ID, like patient 1, patient 2, whatever, and then they have a link to their lymphoma database, which has the additional information. So if a user wants to, and they have the appropriate access, they can see who patient 1 or patient 2 is without that information being in here. But like I said, we'll have a version down the road that will be for clinical data. In fact, that's one of the things we've actually gotten funded from the government, is to work on tools for drug discovery, tools for a platform for cloud analysis, and then a platform for clinical. And then the last thing, I know it's time, but these are going to be really useful to you guys. Again, our idea of trying to get flow accessible to the community and giving people resources to be able to do these types of experiments easily. And so we've worked a lot with BD, with DVS, with the Nolan Lab to actually kind of put the data that's being generated here available to you in a variety of different ways. And the way we're doing that is really using the idea of Cytobank as a platform. So you don't even have to know that Cytobank's running in the background. You can get to this data and be able to work with it in a couple different ways. So the first I'll show you is this site, cytobank.org Nolan Lab. So the protocols you guys are using today are actually going to be available on here if they're not already. Along with that are other protocols that the Nolan Lab has, as well as publications that have come out of the lab. We have something called Cytobank Reports here. And again, we'll talk a little bit about some of that tomorrow. But the idea here is that you can get to a web page where you can see kind of a condensed summary of the data along with live links in Cytobank. And so this, for example, this is the science paper from Sean. You can just go to this website, see what the results are. Then if you're interested, you can get to the data in a variety of different ways. So this is what we're really hoping people will start publishing their flow data in, again, as a way to make this much more accessible to the community. One of the questions that came up earlier today is like which antibodies work, which don't. So BD has done a lot of work with, because they have like I think 3 or 4 different permeabilization buffers, where you can actually see which antibodies work in which buffer. And so if you go to this website, FactSelect, you can actually kind of get a list of antibodies that we have access to. So this is data from BD. And then as you kind of click on one of those, you can actually see data like this, where for every conjugate available to us, we've looked at live across a variety of different permeabilizations, across a variety of different titrations. So again, that gives you another sense of what's working, what's not working, and allows you to get to that confirmation step as you get farther along. Okay? And then finally, with DVS, we just launched another site where you can actually get to the raw data from a lot of the CyTOF protocols you're seeing out there, as well as protocol sheets and actually how to run the information with the data inside a bank analyzed. So you can look at that as well. Okay. So those are all things available to you. Again, you just have to go there and look at it as you go through it. Okay. So a variety of different links to kind of remember. Again, all of this is in your handouts in one form or another. If you ever have any questions, the easiest thing to do is just email helpdesk@sadabank.org. There's also a chat room that you can come to. So that's if you just go to support.sadabank.org, you can get to it. There's a blog that we are actively updating. So every month we send out a newsletter with a couple different posts, anywhere from data analysis to new resources or new protocols we've put up. The resources I was just showing you are sitting at the sadabank.org/resources. And then Sadabank's been published as a chapter in current protocols in cytometry. So you guys can look at that for more information as you go through it. So to end, I believe, let's see, yeah. So again, just to kind of show you what's happened is while I was talking, Angela actually came in, went through, made another kind of sets of illustrations, and started working with this data as I went through it. And so if I'm interested in to see what she did, I would click on heat maps. That would copy over to something we call our working illustration. And I can go into details again this afternoon. But what would that let me do is I could come in, see what she did. I can look at the gating hierarchy to see if she changed anything. Looks like she didn't. I can come down here. I can see how she organized the data. I can look at my illustration, and I can see, again, now she made a heat map of the data as opposed to an overlay. So again, this is kind of the collaborative type things that you can work with and see what's happening. Okay? So again, I think, okay, I'm only 5 minutes late. Apologize again. But here's my email, and let us know if you have any questions. Thanks a lot.